you've had a few lectures, I guess, about population genetics from Armida. And a few of you I know already know a bit about this field. And I just want to apologize to those few of you because this doesn't work. All right, here we go. The others all work? All right. Okay. Because what I'm going to do today is just be like extremely simple and basic, give a sort of very general overview of models in evolution and population genetics, and to the point where it'll almost be insulting for those of you that have a, maybe not just almost, it may just be insulting to those of you who've heard this before, so I apologize to those of you, but still I think it's sort of useful to really step back and look at the structure of models in this area and the kinds of questions that I think are sort of interesting to work on. So what I'm going to talk about sort of broadly over the course of 4 lectures is a few different topics in evolutionary dynamics and population genetics. We'll talk about really sort of some simple theoretical models of evolution. And the first thing I want to do is just emphasize this word population. Because sort of underlying this word, really, what I'm doing is sort of drastically circumscribing the scope of the kind of question that I'm going to be talking about over these 4 lectures. So a population we can think of as some sort of group of individuals, obviously, that sort of interbreeds and that competes with each other for resources in some way, competes with each other for reproductive success. And there's a sort of whole big sets of open questions about what defines a population, how populations are created and maintained, how different populations interact with one another, all sorts of questions like that, which, broadly speaking, we can think of as ecology. So somehow ecology defines what a population is. And we are not going to talk about this. We are going to instead assume that somehow there's some ecological niche or whatever for some set of organisms, and they form some population, and this ecology sets something like a population size. It's sort of, there may be interactions between populations that we're not going to consider, and we're just going to look at the sort of internal dynamics of this population whose structure is set entirely externally by ecology. And I think one of the sort of really interesting directions in evolution and population genetics right now is breaking down this barrier between ecology and evolution. Because sort of traditionally what people have sort of assumed is that ecology is fast and evolution is slow, right? And so the idea has been that somehow because there's some separation of timescales, we can think of evolution as occurring within sort of well-defined populations because ecology sort of keeps everything nice from the point of view of evolution on the relevant long time scales at which evolutionary change occurs. And I think one of the things that's becoming clear, particularly in microbial and viral populations, is that this is often actually not true. And so we often do have to think about those interactions. But I'm not going to do that for the next 4 lectures, okay? But that's a whole other research direction which would be interesting to talk about. All right, so very, very broadly speaking, what is it we want to understand? So the population is, of course, composed of individuals, and they have 2 properties. So they have genotypes and phenotypes. Each individual has a genotype and some set of corresponding phenotypes. And what we are interested in doing somehow is characterizing 2 sorts of properties, right? So we're going to look at specific quantities in a few minutes that are examples of this, but sort of in a very general sense, the kinds of things I'm going to talk about can be divided into the study of divergence and diversity. Okay, so divergence basically describes sort of long-term evolutionary change, right? how does this cloud of genotypes and phenotypes that exists within the population, how does its sort of center of mass move over time, if you like, okay? How do you accumulate what we call fixed mutations that, you know, that in the long term everyone has? So that's divergence. And then diversity is something about the sort of internal statistics of this cloud of genotypes, okay? So somehow at any given moment, any population is going to have some sort of cloud of genotypes and their corresponding phenotypes, and there's some internal statistics of that cloud, and that's something that we can and do observe, you know, and it's becoming easier and easier to observe, and so we want to understand what the expectations are for these kind of statistics, okay? And we'll talk about specific examples of diversity statistics that I'll focus on in a few minutes, but there are many, many things that one could do. So this is a sort of school for condensed matter physics, and I started as a condensed matter physicist, and I think like a lot of the kind of intuition that people have from statistical physics is really useful in understanding these questions. It's why I got into these. But at the same time, I think there's a lot of people that come from physics and really flame out dramatically. And it's sort of important to realize, like, a lot of these questions are sort of operating in fundamentally different regimes than we're commonly used to thinking about in statistical physics. And so the basic issue is that often people want to sort of write down, physicists want to write down kind of mean field type dynamics of evolutionary systems. It's often kind of a natural kind of analogies to equilibrium statistical mechanics present themselves. And that is almost, not always, but often can be unproductive in thinking about these problems. And basically the reason is that we are not concerned with some sort of long-term sort of ensemble average of the genotype distribution, okay? And the basic reason for that is because the number of genotypes that exists at any time, and certainly that even that has existed ever, okay, is really, really small compared to the genotype space. So we are not concerned with the sort of mean field dynamics, rather we're concerned with sort of a more non-equilibrium process of the genotypes and, that exist at any given moment and how that cloud moves, right? So just to, put a scale on this, does anybody have a sense, does anybody know like how many organisms have ever lived? Anybody have any idea like what kind of numbers we're talking about here? Let's just say like how many cells have there ever been on this planet? Anybody have any idea? No, way more than that. There's way more than 10 to the 20 cyanobacteria right now. There are people, I mean, obviously people argue about this. People would say that there's, 10 to the 30 something organisms, cells alive right now. How many have ever lived? Yeah, so that's a pretty good estimate, right? I mean, we obviously have no idea really, but like 10 to the 45-ish is like, it's certainly not 10 to the 100, right? It's like maybe 10 to the 40, maybe 10 to the 50. It's increasing every day, so, all right. Quickly too, actually. Anyway, so, that genotype, that is actually, I mean, it's a large number, but it's tiny compared to if you think about genotype space, right? I mean, we have genomes that are 3 billion base pairs, each of us has on the order of 20,000 genes, right? Like even if you just imagine that, there are 20,000 genes that make a human and there's 2 alleles of each gene, then, there's 2 to the 20,000 possible humans. And really that's like a drastic underestimate, right? There's way more diversity than that that's possible. So the genotype space is enormous compared even to, the sum total of what evolution could possibly have explored in the history of life on Earth. And so, you could write down some sort of equilibrium, like this sort of stupidest sort of thing is like, we'll talk about models where all genotypes are equivalent, right? These are neutral models of evolution. So there's, all individuals have the same phenotype, even regardless of their genotype. And then you could sort of imagine that, if mutations go sort of symmetrically between different genotypes, well, sort of in the long term, each genotype is equally probable, which is obviously both true and useless, right? So we want to actually understand the sort of rates of divergence and the statistics of the diversity. Okay, so that is our sort of really very, very, very big picture goal. And so what do we want to do? All right, so I'm going to start talking about what are the simplest possible models that one can write down. Because, basically my perspective on evolution and population genetics is we don't know jack shit. It's sort of it is absolutely,
YouTube.
What?
It's going on YouTube.
This is going on YouTube? Oh man, I'm going to be in so much trouble. My daughter is going to be like, that is not appropriate. All right. We know, it's really astonishing to me how much basic stuff we don't know, even in the context of the simplest possible models. So let's talk about what sort of basic ingredients the simplest possible models of these processes have to include. So the first thing is, right, we're talking about life and death. We need some birth-death process, right? And so, some sort of stochastic model of how individuals die and reproduce, okay? And, this birth-death process, we'll talk about specific examples in a moment, but it's going to have to regulate population size in some way, okay? And we'll assume that, this sort of regulation of population size, it sort of comes from this ecology, which we're not going to talk about. We'll just assume it's a sort of external thing. But in the simplest model, we need some stochastic model of birth and death. And this will, of course, lead, in some sense, to the process called genetic drift, where frequencies of different genotypes just change randomly because individuals stochastically have different numbers of offspring. All right. We obviously need something of generation of variation. Okay, so mutations. So some process by which every time you give birth, you produce some imperfect copy. That's obvious. And then the third major component is some type of genotype dependent competition, right? What's this called? Natural selection, right? So we're going to try to think about the simplest possible models that we can write down of this process. And they'll basically have 3 parameters to them, and that's it. So obviously, there are more complicated things we could add to this. But we're going to need some population size, n, okay? Generation of variation, we're going to need some mutation rate, which I'm going to call big U, which we can think of as, there's genomes that have a bunch of base pairs. The genomes maybe have length L, and there's a base mutation rate mu per base pair. We'll often work in the limit where, L gets large and mu gets small. And so each new mutation arises at a unique site, and then all we need to worry about is this overall mutation rate. But we don't have to do that. Okay, and then we'll have some distribution of selective effects of mutations, basically saying that, these sites, different mutations can happen at different sites. They have different selective effects. And that the fraction of sites that have selective effects S is given by this distribution rho of S, okay? And we'll talk about what S precisely means in the context of these simple models in a moment, all right? So, but this is basically, I think, the kind of ingredients that we need. So, so let me talk, and I'll talk about what All right, so there are a whole bunch of specific stochastic models that include these basic components that people have written down. Let me talk about a couple of them. So how many of you have heard about the Wright-Fisher model? Have we talked about this? How many of you haven't? Okay, how many of you have heard of the Moran model? It's probably a highly overlapping group. Okay, so about half and half. Okay, so these are just sort of specific stochastic birth-death models that, essentially for historical and practical reasons are very widely used. And we'll use both of these as well as a couple of others over the course of the next few lectures. And so let me just define what they are specifically. So the Wright-Fisher model, okay, assumes that generations are discrete. Okay, so the idea is that we have some population of some size n, okay, and each generation everybody dies, okay, and then we pick some new population also of size n, okay, and the way we pick it is it's like drawing sort of balls out of a bag with replacement, okay. So like I basically say I got to pick n individuals for my next generation, so I'm just going to pick up, at random parents from the previous generation, and each offspring is an identical copy of that parent that I happen to pick, except that mutations occur as some Poisson process with rate u, okay? And so you get imperfect copies of the parents, okay? We can incorporate natural selection into this model, okay, by basically saying that we've got a bunch, each individual has a bunch of mutations that have happened over the course of its, the history of its lineage, okay, and we just keep track of what those mutations are. Each of them has some selective effect S drawn at random from this distribution, rho of S, okay, each of those mutations. And then we say the fitness of the individual is given by the sum of the effects of all the mutations that it has. Okay? And so the idea is, each of these S's, it might be plus 1% or minus 3% or plus 5% or whatever. Positive numbers refer to sort of beneficial mutations, negative to deleterious mutations. At the end of the day, you add these all up, you get some number. And so what we do is we say, okay, what is, first of all, the average fitness of the population? And we compute that by looking at all the n individuals and just averaging. And then we say, okay, if this individual has fitness 3% more than average, you give them the 3% boost in getting picked out of the bag from one generation to the next. Or if they have a 5% disadvantage, they're 5% less likely to get picked. It's just a sort of weighted picking, and that's how we can implement this natural selection. Okay. This is the simplest version of the Wright-Fisher model. I mean, we'll talk in a minute about things that are missing from this, okay? But that is a sort of simplest version, I think, that you can write down. The Moran model is another very simple model that's widely used. It's also sort of discrete time, but it sort of divides time more finely, okay? And basically the way it works is, again, you have some population of size n, and you, at every sort of time step, you just pick an individual at random to kill. Okay, bad luck. And then you pick another individual at random to reproduce and replace that individual. Okay, and again, as before, when that individual reproduces, they just make a copy of themselves, except new mutations arise as a Poisson process with this rate, and those might have selective effects. And here you could incorporate selection either as, if you're more or less fit than average, you're more or less likely to get picked to die, or you're more or less likely to get picked to reproduce. I mean, either way around, you could do it. Okay.
Are they the two really equivalent or not?
With these two? Oh, those two. Okay, so no, that's one thing I want to talk very briefly about. It is slightly different, right? If you implement selection as higher probability of dying versus higher or lower probability of giving birth, okay, in the Moran model. And in addition, right, the Moran model and the Wright-Fisher model, I mean, they're obviously sort of a similar idea, but they're not identical, right? Like if you think about the distribution of the number of offspring that individuals will have in this model versus this model, it's different. And that obviously affects the evolutionary dynamics. It'll affect genetic drift and so on, okay? And there's other models we could write down. We could write down continuous time models. We'll talk about some of those. Like you imagine like a little interval of time dt, and there's some probability, proportional to dt that individuals die or reproduce. And we can write down models like that. And they'll all be slightly different. Yeah?
I have a question about more time scale. Will outcome actually be different? Let's say you have a system where you have like a few different types of food, and your organisms are different in terms of how they consume it. So like won't it be the case that like at t goes to infinity, you'll get the same outcome?
No, not quite. So it depends on what you're looking at. And this is a point, sort of a philosophical point that I want to emphasize here. Populations, like I talk with population geneticists, they're always like so damn concerned about factors of 2 here or there and stuff like that. And, there are differences between these models that will affect certain quantities. Order 1 factors will come up all the time, okay? And you could take 2 perspectives on this. One perspective is that we really have to know the birth-death process of the specific population that we are studying in order to answer questions about how it will evolve. And for certain kinds of questions, like, yeah, you indeed do need to do that, right? But I guess my perspective, and the perspective we're going to take in the next few lectures anyway, because I'm in charge, all right, for the moment, all right, is that to the extent that there are quantities that depend in detail on the differences between these models and other models and details of the implementation, right? It's just hopeless. Like, because there's no way in any realistic population that we're no realistic population is going to be any of these models. It's going to be something more complicated, and trying to measure it precisely is going to be pretty hopeless. And so instead, what we want to do is understand what aspects of the predictions we're making are model specific and which are more universal, and focus our attention on the observables that are more universal, okay? And the extent to which we can do that depends on what we're interested in, of course, but we'll try to do that as best we can, okay? And so that'll be important to sort of keep in mind, all right? Okay, so I just want to talk a bit now about stuff that I am ignoring, okay? Because there's like, we can make, and in fact, let's do this. Let's make a list of whole big areas which really could define entire research programs that I'm just going to ignore, okay? Okay? And we're not neglecting them because I don't think they're they're interesting. We work on many of these things, in my lab, and I think it's super, a lot of them are super fascinating directions. But there's a lot to be done even in the simplest models, and so that's why I'm going to ignore anything more complicated. So can people just sort of shout out stuff that I'm ignoring? Yeah, let's come to that in a sec. Space, good. Right? So in reality, right, this population might be spread out through space, right? And you might, have local carrying capacities, and birth and death depends on where you are, right? That can have dramatic consequences. In addition, one could imagine that the selection pressures depend on where you are in space, right? And that could obviously have a big effect, okay? Related to this, okay, temporal fluctuations, right? Like we will talk about, temporal evolution of allele frequency distributions, but what we won't talk about is, selection pressures could change over time, for example. Mutation rates could change over time. Population sizes could change over time because ecological factors are changing over time. We'll neglect all of that kind of stuff, okay? Other things. Yeah.
If you're ignoring time, so what do you get from those models? Like just stationary distributions?
Well, yeah, I'll talk about the kinds of observables that we want to calculate in a minute. we will do, I don't mean we're neglecting time and that we won't think about it. We will think about the time evolution of the genotype distribution, absolutely. But what I mean by this is temporal fluctuations in the model. So, selection pressures that change over time, okay? Or population sizes that change over time or things like that. I mean, you could think about them and you probably should, but we won't.
But if you don't change it, like you just get like the system of the differential equation and you only control the station.
Yeah, essentially.
Basically, like you are solving the equations that A equal to 0, the determinant of A equal to 0.
Yes, but sort of. I mean, the problem is like these models, like often there won't be like a stationary distribution of genotypes. Like I mean, because of the reasons that I suggested, right? You'll have some cloud of genotypes that's always moving, okay? And then we'll be interested in the statistics of that. But we'll come to that. I mean, that is what I'm going to talk about. So, yeah.
I presume you're going to take a homogeneous population.
What do you mean by homogeneous?
Like all of the things that you're looking at will have the same fitness, same mutation rate, same everything.
Okay, so they won't all have the same fitness, okay, because different individuals will have different sets of mutations. But yes, we will assume, I mean, we may talk about some exceptions to this as we go along, but we'll assume they all have the same mutation rate for the most part. We'll assume that, they have the same distribution of effects of new mutations. If you think about it, the landscape is kind of static. Yes, we will assume the population, and we will also assume things like there's no age structure, right? So that like, I mean, from the human population, you can look around and like a 2-year-old is unlikely to reproduce next year, as is an 80-year-old, right? So like individuals are not actually equivalent, but we're neglecting that, so that's a good thing. I mean, a good point. Call that age structure or other kinds of structure within the population. All right, Ahmed, you mentioned something. I might as well write it down. Right? We will talk about this a little bit because I think this is sort of, it almost goes into the sort of simplest model category. So we will talk about it in a minute. But at least so far as I've defined it, we've assumed, you pick out individuals to reproduce and it's like they just bud, And I work on yeast, so that doesn't sound ridiculous to me. But obviously, humans don't bud. In order to reproduce, you need 2 of them, right? And, they reassort their genomes to produce some new genome. So it's not that you just pick one individual and copy its genome. What you do is you actually pick 2 genomes, and then there's some recombination rate. What happens is, right, you get a big chunk of one parental genotype, and then with some recombination rate, you switch and you start getting the other parental genotype, and you switch back like that, right? And depending, different organisms have different recombination rates, but whatever there's some kind of recombination process that means that you're not just copying genomes, but you're kind of cutting and pasting them in some way, okay? Yeah. Yeah. So like, I mean, we could just think of this as, right? This is like a big topic nowadays. Lots of people working on this for good reason. But we're not going to talk about it, at least for a while, okay? And, for example, like if we wanted to make models of the evolutionary dynamics of cancer or something like this, right, we would clearly have to consider these kinds of things, right? Because new mutations that make individuals more fit will cause the population size to expand and vice versa, and that's really critical to the whole process. Here we're just assuming that like even if the population gets more or less fit, the population size is not affected. It's just sort of set externally, right? And so that obviously just limits the kind of questions that we can talk about.
Epigenetic inheritance.
Yeah, epigenetics. We're just sort of assuming. I mean, in a way, one could put epigenetics into the birth-death process, but yeah, we're just going to ignore that. Okay.
We're also assuming a deterministic genotype to phenotype mapping.
Yeah, I mean, that's right. That's right. The stochasticity comes in only in these very defined ways. That's right. I'm sort of, I'm assuming it. Several of these things are getting into sort of a question about, like, what is fitness, right? I'm defining fitness as this sort of simple one-dimensional quantity, right? Like, you get mutations, they're plus 1%, minus 3%, whatever. You can just add them all up, which is obviously ridiculous, right? But, in reality, there'll be sort of multiple sort of different dimensions of fitness. And, this is sort of can create ecology even within our population. When you think of sort of interactions, all right? So, for example, you could imagine a mutation creates a genotype whose fitness depends on the frequency of other genotypes within the population, right? And that's just absent from these sort of simple models, right? Yeah?
One quick question. So when you're defining fitness, what are you defining it relative to?
So again, and this is sort of the context of our very, very simple models, right? We're assuming that the population size is set externally by processes we're not modeling. And so the only thing fitness can really describe is how much more or less likely you are to reproduce than the average Joe, right? Because the population as a whole isn't going anywhere or isn't changing, okay, in these kind of simplest models, right? So fitness is relative to the mean fitness of the population. And if you're more fit than average, you will tend to increase in frequency, and if you're less fit, you'll tend to decrease. That's it, right? And it's just this simple one-dimensional quantity, so we're neglecting cases where, like, you could imagine, A is more fit than B, which is more fit than C, which is more fit than A, right? Not allowed in my model, although obviously that can happen, right, in real life. Okay, so I mean, it's not that I don't think that sort of thing is interesting, but I'm just trying to sort of focus. Yeah.
Does it mean that you don't allow any symbiosis?
Yeah, correct.
So what do we have then?
You'll see there's a lot of basic shit you can't predict even in this model, right? Like, I know, I mean, I understand, like, you're like, what, you're defining away everything interesting. Okay, I mean, it's true. But I mean, the thing that's remarkable to me is I can define away all these complications just so that it's like so brain dead simple, and there's still lots of stuff you can't calculate. Okay, lots of really like foundational stuff, in my opinion. And if you don't agree, then don't work on this stuff. I mean, that's your prerogative as well. But another type of interaction is epistasis, right? Like here I'm imagining each of these sites in the genome has some fitness effect, which is, characterized by some S, okay, drawn from this distribution. And I'm kind of assuming it doesn't matter what your genotype is. If you get another mutation, it's sort of drawn from this same distribution, right? In reality, like, what can happen? You can get one mutation, and now that changes the effects of a whole bunch of other stuff, right? And generally speaking, this is called epistasis. You might get one mutation that sort of opens up or closes off other future paths to adaptation, right? And that might be really important, and we're neglecting that. So in a sense, this is sort of defining, how many of you have heard of a fitness landscape, right? You've all heard of that, right? So this simple model, I mean, it is defining an extremely simple fitness landscape, right? There is one absolute most fit genotype, right? And everything just interacts additively going up there. It's like an extremely simple fitness landscape. Okay? Yeah.
Yeah, I'm sorry, I have one question.
Yeah.
So if you have like this model where you have only one winner,
Yeah.
so I should ask it, but so what kind of biological system are you trying to model?
Well, I mean, you can take various opinions about this, okay? But as we'll see, okay?
I have no opinion. I'm just
So, I mean, basically what I'm trying to do here, in a sense, I don't, so, Ji-Son's in the room, and so maybe I shouldn't say this, but like, because it's not good for recruiting. But what I'm really trying to do is like define boring, okay? I want to define and understand boring really well, okay? And like this is extremely important in my opinion, right? Like, and we'll see, right? I mean, there's a long history of this in population genetics, right? We'll talk in a little bit about, the neutral model of molecular evolution, right? How many of you have heard of that? Okay, not that many. Okay, well, so good. We'll learn something. But you know, the sort of standard framework in population genetics is, and really like the only thing that we really know well, is, you know, the expectations for these quantities about diversity and divergence that we expect to see in an even simpler version of this model where all these S's are 0. Okay? So you know, you're accusing me of being simple, okay? But this is like even more simple than that, right? And that, I don't mean to say that in a derogatory way. Like that is like the core framework then, that it, you know, it turns out you can get surprisingly rich behavior out of even these sort of very, very simple scenarios. And if we don't understand what that kind of boring looks like, then you really have no basis to be surprised by anything, right? And you have no basis for inferring something more interesting if you can't tell me that you can't get that same phenomena within the context of this simple model. So the basic idea is, like, we're not trying to say this is a realistic model of biology, but it will turn out you can get really rich behavior out of this, and it's something we want to, we will have to exclude this before we can sort of say there's something more interesting going on. Right? Because ultimately what you want to do is you want to do inference, right? You want to observe diversity and divergence, and you want to infer something about what happened. And you need to be able to exclude these very, very simple null models. Okay? And to do that, one needs to understand them. But plus it's fun. I mean, that really is the, I think so anyway. All right, one other thing, the last thing I'm going to write down, right? All right. Okay, I haven't really explicitly said this, but I'm sort of assuming, like, you know, simple, and this is sort of gets into this as well, of course, right? I'm assuming a simple model of haploid evolution, right? There's just, you know, every individual has one copy of their genome, and so I can talk about selective effects like this. In reality, right, for yeast, that's often in the lab, that's perfectly valid, but like in humans, you know, most humans are diploid. And so then you have effects associated with dominance, right? Lots of plants are tetraploid or more. And so you can have all kind of complicated effects associated with that, which I'm going to neglect here. All right. So, yeah.
I have a question. So there could also be rare events, right? That you get a mutation that suddenly changes a lot the fitness. So what is being included kind of in this?
That's allowed. You can have mutations. I mean, the rare events are going to be core to everything we do. And you can have mutations that change fitness by a lot. Okay? Although actually, often those are less interesting. We're going to primarily be thinking about mutations that change fitness by small amounts, just because that will be that will create much of the interesting behavior. Okay, but it's allowed. You can have whatever you want there. Okay, yeah.
So the division between the left and the right. So is the left just the things that are easy to mark, and the things on the right are harder to mark?
No, this is pretty hard.
Okay.
It's all hard.
Relative to each other, let's say.
I mean, I'm just saying that, like, I kind of feel like any model of evolution, right? Like, if we're going to talk about evolution, we have to, this is like absolutely minimal. And I just want to point out, like, I mean, these things are, basically what I don't want is you guys to bitch and moan at me about, like, you know, I mean, you can do that, okay, but, that's great. Let's hear it. No. Okay, so yeah, that's important. Like, I don't want to convey the impression that this stuff is not important. It is important. There are whole research directions that you can and should work on here, okay? And if I had 6 months for the course, you know, I'd be talking to a, you know, the room would empty out over the course of that time. But, but I could talk about all this stuff. And I mean, there'd be other people better than me probably for many of these topics, because people work on all these different kinds of things, and they should do that, and there's interesting stuff. And, but I just have to focus. I got 4 lectures. There's only so much that I can do. And so I'm not saying this is unimportant. All I'm just trying to sort of say is that, yes, you guys are all going to be thinking as I'm doing this, oh, well, what about this? What about that? Okay. Yes, I know that there's all this and that, and they are interesting, but I'm just going to focus on really simple things, at least to start with. Okay. And I'll touch on a few of these things here and there, but like recombination in particular, but I am going to sort of mostly neglect them. Okay, just because I have limited, time. Yeah.
I just try to figure out like what are so as far as I understood, like you're not interested in much in dynamics, but we are very shy.
We are going to be interested in dynamics, okay,
Yeah.
but not. Yeah. Let me define. But go ahead with the next part of your question. Yeah. Fixation or extinction, yeah. Yep. Yeah. Yeah. Right. Yeah, so okay, let's get into that now more precisely. Yeah. I agree. That's exactly what I'm going to talk about. What is it we want to calculate, okay? So we have this simple model. Okay. All right. We have basically 3 parameters. We have some population size, we have some mutation rate, and I'm kind of cheating because this is a whole distribution, but we have some distribution of selective effects of mutations. Okay. So these are parameters of our model. All right. And so, as you asked, what is it that we want to calculate? Okay. So observables. Okay. Now, as I'm sure you've heard about, right, it's becoming really easy to observe things in evolution. So we're going to talk about 2 classes of observables, right? Divergence and diversity. So for divergence, there's a lot of things we could calculate, but what I'm going to sort of focus on, okay, exactly as you said, in the long term, in this very simple model, each new mutation that arises, it will either fix or go extinct. Right? Those are the only 2 things that can happen. And so we could calculate what is the fixation probability of a new mutation. In general, that will depend on the selective effect that mutation provides. Beneficial mutations will be more likely to fix, and deleterious mutations will be more likely to go extinct. And we could try to understand what the shape of this function is, right? And this is basically telling you, what mutations do you expect to accumulate in the long term. And in a sense, it's telling you how efficient do you expect selection to be, right? Ideally, evolution would like p fix to be 0 for s less than 0 and 1 for s greater than 0 from the point of view of adaptation. But obviously that won't actually be the case. And so this is sort of telling you about the efficiency of selection, okay? In how well can it tell good from bad? Right? Like, and a lot of people, we'll come to this kind of theme again. A lot of people sort of think of evolution as an optimization process, right? And we're like trying to but it's not really. Like, it is sort of optimizing, but it can't perfectly optimize. Deleterious mutations will fix and beneficial mutations will go extinct. We can compute quantities out of this. There's various derivative quantities. So one I'll talk a bit about is this one. Okay, so this is the rate of change of the mean fitness of the population, right? New mutations arise at rate nu per generation, right? Because there's n individuals. Each generation, on average, each individual reproduces and generates u new mutations. Those mutations have fitness effect s with probability rho of s ds. They fix with probability p fix. And if they fix, they change the mean fitness of the population by s. And so this is just telling you, on average, how quickly is the mean fitness of the population changing. Okay? You could compute other things, like if you got rid of this S, this would just be the substitution rate, like the molecular clock, right? How many mutations fix per unit time. This is how the fitness changes. Okay? These are basically like the two divergence. I mean, this is just a derivative of this. Okay? And then, okay, diversity, there's lots of things we could talk about. Okay? I am often going to talk about single site, single time point statistics just because they're most widely actually observed in practice. Okay? And there's a lot of work on them, but there's lots of other things we can and will consider. So how many of you know what the site frequency spectrum is? Okay, just a couple of you. So, all right, let me actually define something else first. Okay. We haven't called the heterozygosity of a population. So if I were to sequence two individuals at random from this room, okay, do you know how different you'd be? Yeah, very good. Right off the tip of your tongue. About 1 in 1,000, right? 0.1%, okay? Obviously, there's some whole distribution of this, right? Some individuals are more closely related. Some individuals are more distantly related. So some of us would be higher than 0.1%. Some of us would be lower than that. So there's some distribution. This site frequency spectrum is a sort of more general version of that. Let's say I sequenced everybody in the room. There's what, like 50 people in the room, plus or minus, let's say. So if I sequenced everybody in the room, what I would see is, actually at most sites along our genomes, we'd all have the same base pair, right? We're all identical, okay? And we say those sites are monomorphic. But at some of the sites, you know, some of us would have one allele and others would have a different allele, right? Those are what we call the polymorphic sites, okay? And at the polymorphic sites, some of those polymorphic sites, it'll just be like, you know, 49 of us will have one thing and one will have the other, okay? And we'll say that's a rare polymorphism because only one individual out of the 50 has it. Other sites, it might be 25 and 25, right? And we'll say that's a common polymorphism, right? And so we can, you know, these polymorphisms, they can have any frequency, you know, in the case of sequencing 50 individuals, anywhere from 1 in 50 to 49 in 50, okay? This assumes we actually know the ancestral state, which often we do, but not always, okay? And there's some shape, right? The fraction of the polymorphisms that are present that are rare versus common, okay? And, you know, typically there'll be lots of rare polymorphisms because new mutations are arising all the time and quickly going extinct and they never really get to high frequency. And there won't be so many common polymorphisms. But there's some shape to this, okay? And this is something people routinely measure, right? Like you read any population genetic papers, it's just very easy to measure this, and so people do. Yeah?
I guess I'm missing something. Why is this curve not symmetric? Why 49 versus 1 isn't it?
Yeah, so that depends on what we call a polarization of the ancestral state. So here I'm saying the polymorphism, the ancestral state is one thing and the derived allele is the other. And like typically in practice, like so for humans, right? When we do this in humans, we have a very good sense of what the ancestral state is at every nucleotide because, you know, there's like 20 some other, you know, 8 genomes that have been sequenced. And basically, so like what you're saying is, you know, if you and me differ at a particular site and the chimp has my allele, then I have the ancestral allele, you have the derived one, okay, and vice versa. Now, obviously, we can never know this perfectly, but so often we have these outgroups that tell us what the ancestral state is. In other situations, we might not know that, and then we might fold this. Okay, and then it would go up to like, you know, the sample size over 2 would be the most that you could say. Okay, yeah.
Describe like multiple alleles.
So this just describes. So this is one site. Okay, it's assuming it's a biallelic site. Okay, I mean one could imagine a generalization of this for multi-allelic sites. Okay, which do exist. I mean I don't know. Like I mean I've written papers about this. It's kind of silly, right? Like I mean. people assume that they don't exist even though you can see them. Anyways, this one I'm talking about imagines a biallelic site. Okay, one can compute generalizations for multi-allelic sites, of course. It also, it assumes this is a snapshot of diversity within the population at a moment in time, right? Like we sequence now, okay, and we have this, okay. And so we could also imagine, multi-time point statistics, okay. So we will think a bit about these. So these can be like propagators, like given that you see an allele at a given frequency now, what is the probability you see it at some different frequency sometime in the future, okay? And one can imagine, right, this is also about each site independently. We can also think about like multi-site statistics. So have any of you heard of linkage disequilibrium? Right? So it's basically like a measure of, you have a mutation at this site, it's at some frequency, and another mutation at this site, it's at some other frequency. What's the sort of joint frequency distribution of mutations at the two sites? Okay, and how does that depend on where they're located and so on? Okay, so basically what our goal is going to be, okay, and what, yeah, sorry.
Why is it called linkage disequilibrium?
I don't know. It's because some dude called it that 50 years ago. I'm sure there was a good reason at the time. I mean, I don't know. A lot of these things have names. A lot of the names are stupid. You just kind of have to live with it. Okay. So we've defined like really, really simple model. Okay. And I've told you about some, observable quantities that we want to compute. Okay. And so basically the deal is, what do we want to know? I'm going to give you an n, a u, and a row of s, and we want to compute what site frequency spectra do we expect to see, what p-fix of s do we expect to see, okay? Any other quantity that we might have measured, right? And obviously, to get back to the sort of questions that were in the room as I just sort of defining these simple models, the whole point of this is people really do observe these things. They spend enormous amounts of money sequencing populations and observing these things. All right, and we need sort of a framework for understanding what kinds of, if you're going to do inference and sort of look at observed patterns of diversity like this, or divergence, and try to say something about the evolutionary process, we need to understand what kinds of models will lead to what kinds of behavior. So that's what we're going to try to do. And the sort of, amazing thing, and we'll get to some specific, even simpler versions of this, okay? But really the amazing thing is, you might think, that over the course of 100 years of work in population genetics, this very simple model would be understood, okay? But it is just not true in general that we can, write down an n, u, and a row of s and predict these things. You just can't do it, okay? And so we'll talk about simpler versions of this and what we can't. I mean, there are some things, of course, that we can say, and we'll talk about what some of those things are, and then directions about how we're making progress in getting at some of the things that we haven't been able to do so far. Okay. So I want to very briefly talk about. Yeah. Let me just also mention. Okay. Of course, a lot of these things one can simulate, right? One can make a computational model which, of this very simple process and simulate what kind of site frequency spectra you get and so on, okay? And of course, that's making any kind of progress in this area, you are going to want to have such simulations and work on them and think about them quite a bit. It is actually, though, an area where simulations can lead to, and in practice do lead to, lots of misconceptions in practice in the field. I mean, for two kinds of reasons. So one is, there's basically two problems with simulations. One is that there's sort of widespread problems of identifiability in this field, okay? Meaning that, you can often get identical site frequency spectra, for example, in very different models of the process. And so if you have not, because you will not you'll not be able to systematically explore all possible combinations of parameters, one can often get misled. And we'll talk about some specific, by simulations, and we'll talk about specific examples of that as we go along. The other thing is just tractability. Like, this is one area where actually, if you think about simulating human genomes in human-sized populations, right, that's obviously just intractable even in these simplest models, right? If you just think about the memory requirements that would be, needed. And so, obviously what one would like to do is have some analytical understanding of the process, which allows you to then, take simulations on smaller genomes or over shorter time scales and extrapolate in some reasonable way beyond that. Okay? But that's going to often require some analytical understanding. All right, so let me talk now about just some really simple quantities that we can't calculate. And then I'll start talking about stuff we can actually calculate. Okay, these are just a couple of examples, okay? Not necessarily the most interesting examples, but just as I was, trying to think of simple to state things that we can't really calculate. Okay. Here's one. Okay. Call this stuff we don't know. Sorry, you can see that. All right, this is just one example. All right. All right. So we might imagine, all right, let's imagine we think about this model, all right, and I'm going to keep rho of S fixed and N fixed, and I'm going to vary U. And I want to know what's going to happen, all right, to the population. And I'm going to sort of further imagine that rho of S, like most mutations, you start from genotypes that are reasonably well adapted, and so most mutations are deleterious. Yeah?
Sorry, I just what is the model? So far, only
This model, okay? We can call it the Wright-Fisher or the Moran version of this model, not going to matter for what I'm talking about.
is there any representation in form of equation?
No, I mean, why do you need I've defined a stochastic model of the process. Okay, call it the Wright-Fisher model if you want. Okay, it's just a well-defined stochastic process. I'm doing this picking balls out of a bag every generation, making new mutations, picking them weighted according to their fitness. What more do you need to know? I'm serious. If there's something that's not well defined.
Yeah, I have problems with analyzing equation without C.
There's no equation. This is a stochastic process, right? It's like a random walk, right?
Yeah, I'm sorry. For some reason you really don't want to write, so it's fine.
I will write equations in a bit, okay? But what I'm saying here is this is like a balls and bags model, right? Like I got a big bag that's my population. I'm making the new generation every time. I'm just picking in this weighted way. This is a well-defined stochastic process, right? Okay. So, and then what I want to know is, if most mutations are deleterious, okay, and some are beneficial, how does the rate at which, how does this V, okay, as I've defined it, depend on the mutation rate, okay? And so, all right, for very large U, what's going to happen? Yeah.
So then you have more than one mutation per genotype.
Yeah.
So they're going to be
Yep. So you're going to get some kind of mutational meltdown, right? Like at some level, right, like there's going to be some negative v when we get to really large U, right? If U equals to 0, then by definition, okay, v is going to be 0, all right? And because I haven't done what you wanted and really start writing down equations, I can't show this like explicitly yet. We'll get to that over the course of the next couple lectures. But what's going to happen is, all right, as you increase U from 0, all right, you're going to get some positive v, right? This hopefully should be intuitively kind of obvious, right? If I put in mutations really rarely, okay, then for small enough mutation rate, I'm going to get a positive v, and at some point, I'm going to sort of turn over like this, okay, and go down into some mutational meltdown regime. Yeah?
Sorry, I don't really understand what means
Negative v means that mutations are fixing, right? But in a sense, more deleterious mutations are fixing than beneficial ones, or at least weighted by their effects, right? And so, you guys, many of you have heard about that, right? Talk about mutational meltdown and in the context of like quasi-species models and stuff like this, right? This is not really quite the same thing, but right, you're going to get some, at large U, you're going to get some mutational meltdown. So, you might imagine, right, there's some optimal mutation rate where the population adapts the best. There's some mutation rate over here, which is kind of at the balance between adaptation and meltdown, right? And one might want to ask, what do these depend on? Okay? And what I'm telling you, like, this is a sort of question that we can get into in a bit more detail. All right? Yeah.
I didn't follow the argument of like the small, because if most mutations are deleterious, why would it increase when you have like?
Yeah, so that's a fair point. Like I haven't really said enough that you may be fair to be confused on that point. But basically what we're going to expect essentially is the following, that beneficial mutations are more likely to fix than deleterious ones. Okay? And so when mutations are really rare, selection is going to turn out, and yeah, I haven't really got into this well enough. In a sense, this is the whole question that's not understood. But it will in practice be the case that when mutation rates are really small, selection is more efficient. And the probability of fixing a beneficial mutation will be much bigger than fixing a deleterious mutation. And then as the mutation rates gets bigger, you're kind of just overloading everything, and selection can no longer tell the difference. Okay? And we'll get into exactly why this comes up, okay? But for now, it's fair that you're a little confused. I just want to emphasize like how simple this model is that we can't yet predict. And we will talk about this in detail. It's going to take till tomorrow, so you'll have to be frustrated overnight. But we can make this model even simpler, right? I can just think of 5 parameters, okay? A population size, and then instead of having some whole distribution of selective effects, I could just imagine there's some of them are deleterious and some of them are beneficial. And so this mutation rate gets split into the deleterious and beneficial ones. Okay, we'll just sort of say like there's some overall, a better way to put it is, there's a fraction that are deleterious and a fraction that are beneficial, right? And then we have right? And the ones that are deleterious have this effect, the ones that are beneficial have this effect, right? So really only like 5 parameters in this model, and nobody can sort of predict what the shape of this curve is and how it depends on these mutations, right? These effects. Is equal to 1. Yeah. I just meant there are 5 parameters: N, FD, U, SD and SB. Okay. In reality, there's less, right? Because I can scale time and stuff, right? So it'll only be combinations of these parameters that matter, and so it's actually even simpler than I'm letting on. But in any event, right? Nobody knows how to predict what this function looks like. Okay. You might imagine, like, you could just ask, how does U naught or how does U star depend on these parameters? Okay. And some of these effects, like the sign, is not even always clear, right? So, like, we could imagine, right? How does UD depend, U naught or U star depend on SD, right? So one could imagine, for instance, like, as SD becomes very, very negative, okay, deleterious mutations are really bad for you. But if they're bad enough, they're just never going to fix. Okay? So as SD gets really big, then actually, like, U naught can get bigger, right? You can tolerate more deleterious mutations if the deleterious mutations are sort of worse. Okay? On the other hand, as SD goes to 0, right, they'll fix all the time, but obviously if they get small enough in effect, they don't matter. Right? You can fix lots of deleterious mutations as if they really have almost no effect. And so, you know, somehow this U naught, it depends in some non-monotonic way on SD, potentially also on SB, right? It's just not really clear what this depends on. Another sort of interesting thing to me, yeah. The population, yeah.
Spartans throwing in the babies from the rock, right?
Yeah.
And then the population is still very fit. Right?
Exactly. Yeah.
Okay.
Okay. And so, all right. Another thing that I'm quite interested in nowadays, this is what you might call a V equals to 0 state. Okay? If you had this mutation rate, you would have like V equals to 0. Now you could imagine that this is a natural place for populations to go. Okay? Because let's imagine, I've sort of cheated a little bit here. Let's say I had some mutation rate here. Okay? You're going to tend to adapt, meaning accumulate some beneficial mutations. In the context of this model that I've described, okay, what that means is what's going to happen is, okay, I'm going to accumulate these mutations, you know, at these different sites here, and I'm sort of climbing up on the fitness landscape. And now, sort of as I have accumulated those mutations, right now the back mutations, if I accumulate a beneficial mutation, now the back mutation is deleterious, right? And so what's happening is, like, actually in the context of this model, the fitness effects of neighboring genotypes, right, the rho of S is sort of changing in a very trivial way, right? As I accumulate beneficial mutations, my FD goes up and my FB goes down, right? Just because I'm running out of beneficial and accumulating more deleterious mutations. And eventually, like, I'm going to reach some state, right? Whereas you can imagine, right, as FD goes up and FB goes down, eventually, even if you fix U, you're going to get to this balance point, right, where V equals to 0. And we could ask, like, what does that V equals to 0 state look like? What would be the FD versus FB that corresponds to V equals 0? It's just another way of phrasing this. And that, in a sense, is asking, like, how optimal can the genome be, right? Your population is not going to go to a fitness peak. It's going to go to this sort of V equals to 0 state where the deleterious and beneficial mutations are fixing with equal, sort of at equal rates, weighted by their fitness effects. Okay? And that's a sort of natural place to expect populations to be, right? Because, like, in the absence of any sort of thing, and we can ask, like, what does the site frequency spectrum look like in that case, right? That's a natural null model to think about. Okay. More generally, like, I'll talk about even like more simple models. They kind of break, oh, I can't do that. More simple versions of this model, kind of, I start to cheat in various ways and then you guys complain, and but I'm going to do it anyway. So, like, this is the last sort of simple thing. Then we're going to actually write down some equations, all right? Let's say I have a really, really simple model of purifying selection, okay? I'm going to have basically 3 parameters, okay? I'm going to assume, let me add one more, all right? I'm going to assume that there's 2 kinds of mutations, neutral mutations and deleterious mutations. So my row of S, like there's no beneficial mutations at all, okay? There's just deleterious mutations. And they all have the same fitness effect SD, okay? This is obviously like a ridiculously simple and unrealistic model, okay? So I just have 4 parameters, and really this UN doesn't matter. These neutral mutations will just be markers. So really after I scale time, I'm only going to have 2 parameters in this model, okay? And I could ask, in this very simple model, it sort of breaks this thing because this fitness landscape, because obviously as I fix deleterious mutations, now I should be creating beneficial ones like I was just talking about. But in this broken version of the model, okay, we just have these parameters. And I can ask, you know, what is the site frequency spectrum? Okay. And this is a model we will actually talk about in quantitative detail. Okay. So when I get into a little bit of this, you know, we're still working on this. I mean, we just published a paper from my group, I guess last year now, where we published the first analytical predictions of how the shape of this site frequency spectrum depends on these parameters. Okay. And again, like this is not like supposed to be some realistic model of any biological system, but like there is no population on the planet in which there is not deleterious mutations happening all the time, right? This is like absolutely minimal at some level. And so, you know, predicting the site frequency spectrum in that model is a sort of essential kind of null in comparing to data. All right. So I have 15 minutes left today, so I will hopefully begin to like actually get more concrete with some specific equations. All right? Questions on this background? I mean, you've been peppering me so far, but anything else before I move on to some real specific, more specific at least? All right. All right. All right, so now what I'm going to try to do in, 15 minutes today and maybe half or so of the lecture tomorrow, maybe it'll take the whole lecture, I don't know, is summarize briefly 100 plus years of population genetics. So obviously I can't really do a great job of this, but I want to just give sort of a sense of the core model, of the core results. Okay, so if we think about like, obviously, a lot of stuff is known quite precisely over, you know, a long history of work on this. Okay, so where I tried to think of other places to start, but I couldn't really. At least there are probably better places to start, but I can't figure them out. So what I am going to start with is a sort of, you know, if you pick up any population genetics textbook, we start where sort of people historically started with models of mutation, selection, and genetic drift acting on a single locus within the genome. Okay? So we're going to imagine that the genome is of length 1. Okay? And this is obviously, you know, silly, but like this, there's extensive body of work on this which we have to understand if we're going to move forward. Okay? And so basically we're going to imagine that there's one site, okay, in our genome. Okay. Implicitly, what we're kind of doing is imagining everything else can be neglected, which could be true for one of 2 reasons. Either nothing else, selection isn't acting on anything else, or because recombination happens rapidly enough that you're kind of decoupling dynamics at different loci. Okay, and we'll talk about that as we go along. But for now, we're just going to assume we can do this. Okay, now we're going to imagine there are two alleles possible at this locus. Okay, big A and little a. You know, in the old days, like a locus might be a whole gene. Nowadays, you know, we might think about a single nucleotide site. And so in reality, you know, with a single nucleotide site, there should be 4 alleles. Okay, but we'll think about, I mean, just for the sake of, I mean, it's not so hard to include 4 alleles, but all of the sort of basic ideas can be gotten in this simpler model, and this is what people traditionally do. So we'll imagine that this is some kind of artificial site in some genome. It can have 2 alleles, big A or little a. And you know, there's some mutations back and forth. And this guy, let's say, has some fitness S, some fitness effect S relative to that. Okay? So we have mutations, we have selection, we'll have genetic drift within the context of our Moran or our Wright-Fisher model. Okay? And so what is going to happen at this locus? All right? So basically, there's like one thing that we could keep track of, which is the frequency of allele big A, or alternatively of allele little a, okay, at that locus. And so we'll define, okay, X is, let's just say, okay, I have to define it one way around. I forget which way I did it in my notes. I'm going to say there are I individuals of type little a I and n minus I of type big A. Okay, so I'm going to define my X equals I over n, the frequency of the allele at little a. Okay? All right, so strictly speaking, right, this can take on discrete values, I running from 0 to big N. Okay? All right, and what we can be concerned about is computing the probability distribution of having X individuals of type little a at time t. Okay? More generally, like we can do this sort of conditional on having some other distribution of X at some earlier time, right? There'll be some initial condition here, and this is sort of defining how the probability distribution of the allele frequency changes over time. Okay? And so it's kind of obvious if you think about it what's going to happen. Okay? Let's say at some time I have little I over n. Okay? It's easiest to think about this in the context of the Moran model, so that's what we'll do for the moment. Okay? In each time step, okay, what can happen to little I? Right? It can go up or down by 1, right? Or it can stay where it is. Right? What has to happen for it to go up by 1? Just as an example, right? There's a few different ways it can go up by 1. We have to kind of list them all, right? You can either pick let's see, pick a big A to die, pick a little a to replace that individual, and that little a doesn't mutate. Right? Or you can pick a big A to die, a big A to replace that one, and the big A mutates to little a, which occurs with rate mu. Right? So if we think about it, right, the probability, okay, in a single time step of going from I to I plus 1, all right, we need to pick a big A to die, okay, which occurs with rate, okay, let's imagine, for the purposes of doing this, I'm going to imagine S equals to 0, just because it'll be easier to write down. We can then think about what happens when S is not 0 in a minute. Okay. Pick a big A to die. Sorry, that occurs with probability n minus I over n. Pick a little a to replace it, and that little a doesn't mutate like that. Okay. Or I can pick a big A to die and a big A to replace it, which occurs with this probability, but that big A mutates and becomes a little a, right? I get confused writing these things down the closer I get to the board, but I think this is, if I've not forgotten anything, these are the only two ways I can go from I to I plus 1 in one time step. Shout out if because I will like drop shit. All right. Now, we could include selection in this, okay? By basically, what I can do is I can define the mean fitness of the population, okay, as essentially okay. This is saying type big A has fitness 1 plus S and type little a has fitness 1, and then the probability would go like this. All right. Okay, if we assume that fitness works not on death, so everybody's equally likely to die, but the people that are chosen to reproduce, the individuals that are chosen to reproduce are chosen in this weighted way. Okay, the probability at little a is chosen to reproduce is just 1 times I over n times W bar. Okay. This is just sort of weighting individuals. Okay. I've just kind of weighted individuals by 1 plus S if they're type big A and by 1 if they're type little a. Okay. This is just a definition. This is what I mean by S, essentially. This is the definition of fitness, if you like. You could define it in other ways, like we were talking about before. Okay. All right. So we can write down the probabilities. Similarly, this is I to I plus 1. You could write down the probability of I to I minus 1 or I to I, right? And those are the three things that can happen in one time step, right? So from this, what one can do, I mean, what you can see from that is, like, this is going to be a kind of a biased random walk, right? You start at I, you have some probability going up, down, whatever, right? There's some bias due to mutations and selection, and there's some diffusion due to random genetic drift, okay? And here, like, the physics and PopGen words become contradictory. We'll get to that in a sec, right? But you can see from this you could write down some master equation which would describe the probability of having I individuals of type little a at time t, okay, and how that depended on having I plus 1 or I minus 1 at some one time step earlier, okay? And in the kind of way that's like totally standard and all kinds of problems like this in physics, all right, what you will find is, you know, if you take a limit, okay, and this is again a standard limit will take a lot. If n is reasonably large, we'll treat both x and t as approximately continuous, okay? So really t is like these discrete time steps, and there are like big N, in the Moran model, there are big N time steps for one sort of generation, right? Because on average, I mean, the definition of a generation is on average everybody dies, right? So n time steps is like one generation. And so if n is large, the sort of time steps are sort of close together, we can treat t as approximately continuous. And so we'll do that, and we'll just measure time in generations. And similarly, we can treat x as approximately continuous. It's not necessarily obvious we can do this right near 0 and 1, okay? And whether or not we can depends on the application. But for this, it's pretty good for what I'm going to tell you about. And what you get then is you get some diffusion equation. I'm just going to write down. Okay? All right. So you can kind of, I mean, this is a good exercise actually, right? I've told you what the Moran model is. You know, we computed one of these probabilities. You know, by analogy, you should be able to sort of write down the other ones and write down the master equation, and this is the sort of equivalent Fokker-Planck equation that you get, okay? And it's just a sort of diffusion equation for this allele frequency x, okay? So, which should be unsurprising since it's a sort of random walk, okay? This is what we call genetic drift. It's the sort of diffusive term, but in population genetics, we call that drift. Okay? And these two terms are what a physicist would call drift, but are called in population genetics, this is called selection, right? And this is the mutations, okay, for obvious reasons. Okay? And we'll come back to looking at this equation and what its implications are next time, because I'm almost out of time. But here, like for the last 2 minutes, I just want to point out, like, you can sort of see from this equation kind of what's going to happen. All right? First of all, I mean, let's just look at this genetic drift term. I mean, you could imagine that S and mu and nu were 0 for the moment, right? If we just had genetic drift, what would happen is, I mean, you just get diffusion, right? And we get this sort of allele frequency distribution. If you start from some fixed allele frequency, then over time there's sort of random effects, and so the allele frequency distribution kind of spreads out. You have some probability. It spreads out in some roughly Gaussian way, okay? But, okay, there's two things. First of all, the rate at which this happens is inversely proportional to n, right? So genetic drift is stronger in small populations, which makes sense. Because there's sort of, genetic drift is just like counting error in the birth and death, right? So it's natural that there's this sort of effectively square root of n dependence, okay? However, it also depends on the allele frequency. And that, if you think, that's sort of what makes this kind of diffusion equation a little bit more complicated than like a standard diffusion equation. Because like normally, like this would be the diffusion coefficient, okay? But you see the diffusion coefficient depends on x, okay? And so what kind of happens is, right, the diffusion is strongest for x equals a half, and it gets sort of slower near the boundaries, okay, which kind of makes sense, right? Because in order for you to change the allele frequency, you have to pick one thing of one type to die and the other thing of the other type to reproduce, okay? I mean, absent mutations, okay? Mutations will obviously do other things, but without mutations, you just have to get, you know, one of the guys to die, the other guy to reproduce, and so it's, you know, that's going to depend on x times 1 minus x. So what's going to happen kind of is, like, let's say, let's not start in the middle, okay? Let's say you start over here somewhere, okay? You kind of diffuse around. It's sort of initially Gaussian, but then it sort of starts broadening out more this way than this way, right? And you can, you know, you can sort of intuitively figure out what's going to happen. And then, of course, you know, selection is going to act to sort of, you know, move this whole direction in some, move this whole distribution in some directional way, okay, towards the allele of higher fitness, of course. And then mutations are going to sort of act to sort of push in from the boundaries, right? And that's what these things are doing, okay? And so these things all also, you know, these coefficients depend on the frequency, which sort of makes sense why they do, and you know, it's not like some deep thing, but at the same time it does actually make the solutions to these diffusion equations in population genetics a lot sort of messier than the solutions of sort of traditional diffusion equations in physics. But you can solve these, okay? And basically, you know, there's a whole sort of series of papers, I guess in the 50s mostly, in the 60s by Kimura and many of his collaborators and other folks, actually, you know, solving these diffusion equations for various different values of S and things like that, various different scenarios. And so we'll get into, you know, what some of those things look like and the implications in a bit. But the bottom line is that if we just have sort of single sites, Okay, we can write these things out very, very well. Okay, we can solve them. And so to the extent that we can think of genomes as just collections of independent loci that all evolve kind of independently from one another, the problem is basically solved. Okay? I mean, I haven't solved it here, but people have solved it. Okay? But as we'll see, like that is not actually a good approximation in general, and we're going to think about, about what's wrong with it and so on. All right, so I'll pick up there tomorrow.